Good evening. Welcome to the Creighton Darwin Day celebration where we hope to light up your brain. This is our new banner that we just ordered. It should be here in time. It will be here in time for today. So reason is rationalists, empiricists, and skeptics of Nebraska, for those of you that don't know. Our mission is to encourage critical thinking. Our members are dedicated to researching and exploring fallacies in such areas as medieval quackery, pseudoscience, Bible literalism, religious and political extremism, racism, and homophobia. We promote the use of scientific methods, evidence, logic, and reason to gain insight and understanding. I'm Lori Steinke. I have the honor to be the president of Reason at the present moment. Les Lane is the vice president. Jim Bechtel is the treasurer. And we want to welcome all of you to our 16th Annual Darwin Day celebration. This is a very special day for me personally because 10 years ago I came to my first Reason Solstice party, and then 10 years ago Chuck Austerberry was giving the Darwin Day presentation, and I came to my first big Reason meeting where I met a Reason member who I ended up marrying. So I am very grateful to Reason. I am very grateful to Chuck, and this has been an absolutely wonderful group as far as I'm concerned. So tonight we have Chuck Austerberry, assistant professor of biology from Creighton University, to speak for us. He's the director of pre-health professions advising there. Chuck got his education at Kalamazoo College, where he earned a BA in biology, graduating magna cum laude, and a Ph.D. in molecular biology from Washington University. He did research assistantships and graduate fellowships at Michigan Cancer Foundation and Fred Hutchinson Cancer Research Center before taking a position at Creighton in 1987. He's a member of the American Scientific Affiliation with the Affiliation of Christian Biologists section. He's also a member of the National Center for Science Education, the Nebraska Academy of Sciences, the Nebraska Religious Coalition for Science Education, of which he is a co-founder, Sigma Psi, the Scientific Research Society, and the National Association of Advisors for the Health Professions. Chuck has given lay and scientific talks over the years that have been very enlightening and very accessible both. I really appreciate the way that he can explain molecular biology to a lay audience. I'm very pleased to have him here with us tonight. His talk is entitled, pardon me, I need to get back to the beginning here. Valuable Scrap, How New Genes Can Evolve from Odd Old Pieces. You join me in welcoming Chuck Austerberry.
Sounds like this is working. That was wonderful. Thank you. Very kind, Lori. And what a treat to hear about sort of the anniversary of sorts that this is. That's a real neat thing. And it is a pleasure, actually, in so many levels for me to be here. It's hard to believe it's been 10 years. When I talked last time for this Darwin Day celebration, as I recall, I was talking about DNA, but it was more in its role as just evidence for phylogenetic relatedness and the evolutionary history, the ability to use that evidence to draw phylogenetic trees, kind of mapping out that history of bifurcation replication and speciation over the eons of time. I was really delighted to be able to choose my own topic again, and this time what captured my interest, and hopefully will be of some interest to you, will be how new genes form. And one of the things, or many things that's so interesting about how new genes evolve is how they kind of reuse and recycle old parts. And sometimes those old parts of DNA are just fascinating. The way genomes have found to put use to those parts, very different perhaps than their original function. So I want to just do a quick review of DNA. Now, for some of you, you work with it every day. And for some of you, it may have been a few years since you learned about DNA in school or something. So just to kind of put everybody on the same page, remember that deoxyribonucleic acid, or DNA, is a polymer, a string of repeating units, and we'll call those repeating units monomers. The building blocks, as we put on this slide. And each of those building blocks is called a nucleotide. And a nucleotide has the three parts you see here. So a nucleotide has got to have a phosphate and a sugar. In the case of DNA, that sugar is deoxyribose. And it may have more than one phosphate, but at least one if it's a nucleotide. And then it's going to have a base. And these bases, this one, G for guanine, it is important in the sense that it's the base that distinguishes one nucleotide from another. So if you're looking at a DNA molecule, you're going to be seeing a sequence of these nucleotides, and the backbone part, the sugar phosphate part, is going to be the same, it's uniform, but what distinguishes one nucleotide from the other will be the base. And you see the guanine, cytosine, adenine, and thymine labeled here. Now we're looking at one strand of DNA, but as you know, DNA is double-stranded, and the two strands pair with each other with a type of bonding called hydrogen bonding. As you might even see from this diagram, there's two of those for every pair where thymine and adenine are pairing with each other, and there's three of them for every pair with guanine and cytosine. And then that's kind of the pairing rule. It's G with C and A with T. You've probably seen the double helix also in this, in this helical form rather than the flagged out ladder form. And indeed, Watson and Crick shared the Nobel Prize along with another individual, and there should have been some more people, Rosalind Franklin included. She survived. But that was quite a discovery to get that structure, because the structure serves the function in such an obvious way. So if you want to copy the genes, you want to copy the DNA and preserve that information across cell generations when a cell divides, and across organismal generations when parents have children, for example. And so that information is maintained because of those pairing rules. The strand on the bottom we refer to as the template strand. The strand on the top is the new strand that's being synthesized, and which of these four types of nucleotides is going to come in next to elongate the 3' end of the growing DNA strand is going to be determined by does it pair with the partner in the template strand. So, double-stranded DNA is replicated in a mechanism we call semi-conservative. All that means is that the two parental strands separate from each other. Each serves as a template for a corresponding new strand, and voila. Now we've duplicated this double-stranded DNA to make two identical daughter duplexes of DNA. Now, there are sometimes changes that happen during DNA replication, and there can be other events that can change the DNA. And thank goodness, we wouldn't have evolution if it weren't for those changes, those mutations that can happen, those copying errors and other things. But it's amazingly accurate and faithful, and actually Now, the DNA we know is what our genes are made of. It's got the information, but it doesn't do any good unless it's what they call expressed. It's got to be read out and do something. And for DNA to be expressed, it needs to be transcribed. So if you think of a transcript is, like a legal transcript might be a written record, or probably a digital now, record of a court proceeding. So maybe the words originally were spoken out loud, but now they're in a form, same language, just a different form. That's what transcription is. It's the same language in the sense of another nucleic acid called RNA, and then R stands for ribose, whereas the D in DNA stands for deoxyribose. There's simply an extra oxygen in the ribose of RNA that is missing from the deoxyribose of DNA. But other than that, that sugar phosphate backbone is virtually the same. So don't be fooled by this blue color here. It's really very, very similar chemically. Now that extra oxygen does make a lot of interesting differences, but it's still a nucleic acid, and I think it's appropriate that making a RNA molecule using the template strand, one of those two strands of DNA will now be the template to make the RNA with the right sequence of bases, the right information. We call that transcription. But a lot of our genes don't code for a functional RNA in the sense that that's the final product. That transcript, that RNA, is going to be translated. And that really is a good word too, because we're really converting the information now to a different language, if you will. Instead of a sequence of nucleotides like DNA or RNA, we're going to make a polymer, a sequence of amino acids. And we're going to call this a polypeptide or protein. And there's a wider variety of amino acids to choose from. The most typical set of amino acids that our proteins are made of, it consists of 20 different amino acids. Whereas the nucleotides that make DNA or RNA, there's a set of 4 for each. In fact, overlapping set for 3 of the 4. But it doesn't matter really in the sense, you might think, well, proteins are a lot more complex. Chemically, and the conformation shapes that they can fold up in, certainly. The jobs they do, certainly. But, computer language tells us that all you need is 2 different options, right? And you can have a binary code that can still, in that language, store and then have that information be used and expressed. So it works very well. So when we talk about new genes evolving, what are we talking about there? And let's kind of just think a little bit about what we mean by a gene. And that's actually not as easy to define as you might think. I mean, I think we all have kind of a sense of what a gene is. Here's a definition I like. A segment of DNA that's expressed to yield a functional product, either RNA or a polypeptide protein, that would be made by translating an RNA. Now, in our genome, as humans certainly, there's a lot of space between typical, the distance between genes is typically quite long. And let's take a look at what a gene in an organism like us, an organism that has cells with a nucleus in each cell, eukaryotes as they're called. So in the nucleus of our cell, we've got genes spaced out typically pretty far from each other. And then what we're looking at in these colored sections would be parts of the DNA that get transcribed. And this would be what's called a primary transcript. So that's the RNA, kind of a one-to-one correspondence with the segment of DNA that was the template for that transcript. And then notice this mRNA, this messenger RNA down below. And it's missing these 2 blue segments corresponding to these introns in the DNA. One of the things that is typical of the genes in eukaryotes is that they are interrupted with these introns. And so there's not only spacers, if you will, from one gene to the next, between genes, there's also spacers, in a sense, within genes, these introns, these portions that get copied into RNA, and they're part of the primary transcript, but they're going to get spliced out to make the mature functional messenger RNA that can now be translated to give the protein product. So again, the entire gene is transcribed, introns are spliced out, exons are what's left in the mature messenger RNA. An average human protein coding gene contains about 9 exons interrupted by 8 introns and about 30,000 base pairs long. It's just an average. There's a wide range, many genes much smaller and larger than that. Now, that splicing we're finding doesn't always happen exactly the same way. So the same gene being transcribed multiple times to give identical primary transcripts can actually give rise to a variety of proteins when different messenger RNAs are formed by what's called alternative splicing. So if you look at these 3 messenger RNAs, each could come from the same kind of primary transcript, but the splicing pattern can differ, and you can get different protein products as a result. That's one of the ways that our genomes have much greater potential for coding a wider variety of proteins than if you just assume that each gene could only code for a single kind of protein. All right, so our topic tonight is how do new genes evolve? Now, if you're asking, well, what do we mean by a new gene? We're talking about a gene that would appear in a species or a group of species, but not others that would be evolutionarily related and would share most probably of their other genes. So here we see a phylogenetic tree of primates, and you see that there are a bunch of these orthologs, genes that are homologous across these species, but sometimes you'll find a DNA sequence in a genome, and you look at that location in the others that are related, and you don't see that gene there. Now, there's a lot of possibilities for this. One would be that maybe the gene has been lost. But of course, you'd have to be arguing here that if it's, say, only in one species and not in the genomes of all these others, you'd have to be arguing that it was somehow lost from all of these others, that they originally had it in their history, in their lineage, and lost it. So, it could be that maybe it's just very quickly evolving, right? And so this stretch of DNA doesn't resemble this anymore. Wasn't necessarily lost. You're kind of arbitrarily talking about one being lost and the other not, but there's just such difference that you don't see the similarity anymore. That's a possibility as well. But I guess what I could be talking about would be neither of those, where indeed an event happened at some discrete point in time, and a new gene appeared at a location where previously there either was no gene or there was a totally different kind of gene. At least that's what I'm coming interested in. And even that can happen in a number of different ways. So first I'm going to look at some examples with you where genetic material is coming in from outside the organism, okay, in the form, actually, of a virus. Right? Viruses have nucleic acid genomes, either DNA or RNA, and a lot of viruses infect our cells, go through their life cycle replicating, making more virions to infect more cells, but they never kind of leave any part of their genetic material to become a permanent part of the host organism genome. However, there are other cases where a virus does just that. And we'll look at some interesting examples where that's happened. Certainly, lots of cases are known of new genes arising from gene duplication. Right? So there wasn't previously a gene over here, but the gene that we see there arose as a copy of another gene in that genome that served as kind of the original master copy, if you will, and then a duplicate was made. And once you duplicate a gene, especially if one is enough to do whatever the original job was, you can sometimes have neo-functionalization and other things where there's divergence, and now the new one might serve a totally different function than the old one. And then finally, the last kind of case we'll look at, they call it de novo gene origination. From DNA, the DNA sequence has that AGCT kind of chemistry, but other than that, we're talking about DNA that didn't previously serve a gene function, didn't come from some other copy of a gene in the genome, didn't come from outside, a gene in a virus. It just emerged at that location de novo. And I think we'll look at an example of a RNA, a functional RNA, a gene that encodes a functional RNA, evolving from what previously was a protein coding gene, and then another example different from that. All right, let's start then with that first type, invading from outside, a virus infecting the cells of an organism. Lots of different kinds of viruses, but the viruses that are especially relevant here are viruses that, as a normal part of their life cycle, a copy of their genome is integrated. It recombines with and becomes integrated into a chromosome of the host cell genome. Retroviruses do that. Retroviruses are called retroviruses because they have reverse transcriptase, the enzyme that takes RNA, which is the form of nucleic acid that they carry in a virus particle. When they infect a cell, that RNA gets reverse transcribed to DNA, and a DNA copy of their genome, double-stranded DNA, is integrated as what we call a provirus in the host cell genome. And that DNA can be transcribed. You can get RNA products, and some of those RNAs are new genomes to be packaged into virus particles. Some of them are messenger RNAs to be translated to produce the viral proteins. And typically these kind of viruses, by the way, don't burst the cell open. They kind of just bud off, taking a little bit of the surface membrane with them as they leave. Now here's a little close-up of this is probably meant to depict the HIV virus. But the focus here is on these envelope glycoproteins. So these types of viruses have not only proteins surrounding the RNA genome, but they have a membrane, very much like our own cell's membrane, a lipid-based membrane surrounding the virus, and in that would be these envelope glycoproteins. They interact specifically with the host cell surface. In fact, there's usually a specific interaction between an envelope glycoprotein of a retrovirus and a particular cell surface protein or proteins that act sort of like receptors. It's not that the cell evolved these for the sake of the virus to invade it, but the virus has evolved to exploit some molecule that cell has on its surface, maybe a solute carrier or some signaling receptor or something, and that virus has evolved to exploit that as its attachment point, its landing pad. And then these envelope glycoproteins also help mediate the fusion of the envelope membrane surrounding the virus and the cell's plasma membrane, the surface membrane of the cell, and that allows the entry of the core of the virus with its genome. Now, once that viral genome gets in, does the reverse transcription, the proviral DNA is integrated, it can be expressed, transcribed, if it's messenger RNA products translated. Some of those proteins, those viral proteins, will end up on the surface of the cell, including, of course, these envelope proteins, because that's where they need to be to get picked up as the viral capsid buds off and takes some of the surface membrane with it. Okay? Now, why are we talking about some of the details of a retrovirus life cycle? Well, it's because when I want to talk about an interesting example of new genes, I'm going to be talking about genes that come from these retroviral envelope glycoproteins. Glyco just means sugar. These proteins typically have some sugar groups attached as well. Now, the HIV retrovirus is more complicated than this one shown here, but a typical sort of stripped down simple retrovirus really has these three genes. The GAG gene encodes some of those core proteins. The POL gene encodes that reverse transcriptase enzyme that takes the RNA template to make a DNA. Also the integrase that does the recombination event inserted into the host cell genome. But it's the envelope glycoproteins that enable the retrovirus to fuse with the host cells we saw. And another thing they do is they suppress host immune responses. Well, what good would a protein that facilitates fusion of membrane and suppresses the host immune system do for, say, a primate? Well, turns out that some of those retroviral envelope glycoproteins have been put to good use in a placenta. Because if you think about it, a fetus in the womb of the mother needs to avoid an immune reaction against the antigens of that fetus that would be encoded by the paternal genes, different from the mother's own. And so that fetus could be recognized as foreign. We don't want, an immune response attacking the fetus. And it turns out we also do want some fusion to occur between, not in this case a virus in a cell, but between different cells. Now, how does this kind of work? So here we have our proviral DNA. When a provirus first enters the genome, it's probably, in many cases, most cases, has all its genes functional. But what I'm talking about now are these endogenized relics, if you will, of long ago infection. And if that infection has happened in the germline, then it can be passed on from generation to generation, just with all the other genes of the genome. Vertical transmission of offspring, in other words. Now, if it was recently entered into the host organism, it might still be actually producing some infectious virus particles, because it would still have all three of those essential viral genes to do that. And there are certainly, at least in mouse, some examples like that. A mouse memory tumor virus, leukemia virus in mouse as well. But most of what we're looking at now here would be cases where maybe the only gene that's functional anymore is a derivative of the envelope gene, the gene encoding that surface glycoprotein. And it might have been kind of combined with some other genetic material that's in the host genome. By the way, these LTRs stand for long terminal repeats. They're at the same, they're at the ends of the sequence, and they're similar to each other. They're very strong in promoting expression protein. So let's say the GAG and the POL gene have been mutated, they've decayed, they're no longer functional, no longer encoding functional viral proteins, or any protein probably. But we have a transcript, and that material can be spliced out at the RNA level, and you can get a functional messenger RNA that has that derivative of the envelope gene. And we call these genes that serve a role in the placenta I'm thinking of here syncytians. Because a syncytium, or syncytia plural, is the word we use for something that happens when cells fuse. Okay, here we have two cells. And if they fuse, now we've got a multinucleated cell. It's got more than one nucleus. In this case, two. You could have this happen multiple times and get more nuclei that are no longer separated from each other by being surrounded by an individual cell's membrane. Rather, they're sharing, they're in the same shared cytoplasm environment because they're in this syncytia. So it's very clear, there's good evidence that the glycoprotein on the surface of endogenized human retroviruses have provided kind of the raw material for these critical fusogenic, as they call it, proteins that enable the fusion to happen. Here's a little map. You'll see maps of genes that look like this, kind of a long bar. So this has got a few different parts. This ISD stands for the immunosuppressive domain. Some of these still retain that ability, some have lost it through mutation. The yellow part, the transmembrane domain, is the part that anchors this protein in the surface membrane, the cell membrane, the plasma membrane of the cell. And so it's got most of this part of it outside the cell. In fact, there's actually a cleavage site. There's a cellular protease that cuts this protein into two parts, and the parts still kind of bind to each other and stay together. This surface part, SU, is what's indicated here, and then the rest of it, the transmembrane part, is shown by the thinner line with its immunosuppressive domain indicated as well. And the RBD just stands for receptor binding domain. So like we saw, for this thing to function, it's going to be binding to kind of a partner on another cell, a receptor, and facilitate that fusion. So definitely, if you're looking at cells that might otherwise be unable to fuse, if you kind of experimentally introduce the syncytin genes, you can get those cells to fuse and form syncytia. Now, in the mouse, we know that it's syncytin genes that have evolved from endogenous mouse retroviruses are essential, because you can do what they call knockout. KO stands for knockout experiments, where you see what happens when that gene is no longer there, no longer functional. And what happens is you don't have viability. You don't get baby mice. That's especially true with one of these more than the other, but you do the double knockout where it's missing both of them, and they die very early, actually, during the gestation in the mouse. In fact, it's interesting, when a mammalian fetus, the placental mammal type, like mice or us, develops, the placenta is derived from the outer layer of the early embryo called the trophoblast, and the syncytiotrophoblast refers to these multinucleated tissues that form through that cell fusion. In the mouse case, not the human or primates, but in the mouse case, there are actually two layers of the syncytiotrophoblast, and one of these two syncytin genes is expressed in one and another in the other. But what this is, of course, is where you can have nutrition and gas exchange and all occurring between the fetal blood and the maternal blood. Very essential for a viable pregnancy. If we look across some different mammals, type of found the mouse, we said, is a little different than human, but actually, both mouse and human share a feature of there really being nothing separating the maternal blood, the mother's blood here on the left, from the fetal cells and the fetal blood system, other than this syncytial trophoblast layer. So this is called hemochorial, blood chorial. The chorion is just exposed right to that blood. Now, in the carnivores, there's a little more separation. There's an endothelial cell layer. Endothelial cells are the ones that kind of line our blood vessels. And so there's still that there, but otherwise, it's very much, so here's the maternal blood still inside of blood vessels, and there's the fetal, but there's this syncytium between them. There isn't another layer of cells. Now, when you get to the ruminants, like cows and such, here we have a synepithelial chorial kind of structure where there's a little more separation, these epithelial cell layers. But even there's been endogenous retroviral-derived syncytium genes discovered. The only type that we don't see them in are some mammals like pig and horse, where you just have these regular flat cell layers, or epithelial layers, one on the fetus side and one on the mother's side, and there's no kind of syncytial system in the placenta between them. I mean, there's a placenta, but there isn't that same kind of intimate connection that we see in the others. So when you do have that syncytial of some sort, trophoblastic layer in the placenta, apparently it's always because of these retroviral envelope glycoproteins providing that fusogenic function. So here's some DNA sequence right where this endogenous retrovirus that gave us the syncytium 1 gene is inserted in some mammalian species. If you look at the New World monkeys as opposed to the Old World or the apes or humans, they don't have that particular gene. They do have placenta, but they have their insertions elsewhere. Now, there was one earlier than that gave rise to a gene that I'll call the syncytium 2 gene, although it actually has an earlier history. And that one was inserted prior to the branching off or divergence between the Old and New World monkeys. So that would be in both. Now, on the left, you see a wider range of mammals, and each of these red triangles represent insertions inferred where these endogenous retroviruses inserted and gave rise to genes. A carnivore group that includes dogs and cats was found more recently. Rabbit lineage has their own. There's an African animal that recently was found to have one up in that lineage. It's quite separate from others. Now, you notice you don't see one here for the sorry, there they are, the marsupials down there. But just in the last year or so, that's also been discovered. So this was published actually just last month, late January. So the work was, I'm sure, done before that, but here we have 3 insertions, one very early, and so that one is in common across all the marsupials. One that is in common across all of the opossums, and one that's more recent than that, only a couple of the opossums. And this more recent one, we know it's quite recent in part because it still gives infectious virus particles. The other couple genes in the retroviral genome have not decayed to the point of becoming non-functional. And now going back to that really old one here, here's a comparison of the opossum, Tasmanian devil, and wallaby versions. They certainly have the functional, fusogenic envelope glycoprotein that's going to be needed for that placenta. Now you might be wondering, why would a marsupial need a placenta? Well, actually for a few days, it's only about maybe 10 days or less. Early on, there is a and it's going to be getting its nutrition from lactation. And that's why it's a marsupial meal. But even with the marsupials, very early on, there is that placenta. You'd have to go back to an egg-laying kind of mammal if you wanted to find one that didn't have any kind of placenta. Okay. So a gene coming in from the outside was one example. Now let's take a look at another way that we get the genes. Duplication, unequal crossing over can happen. All right, there's exchange of genetic material that happens. It's very important in meiosis. Well, sometimes it happens in kind of an unequal, abnormal way, and you can end up getting an extra copy sort of in tandem nearby. It has happened a lot. Another thing that can happen, remember we said that if you've got a retrovirus, either recently or maybe long ago, into your genome, then you've got reverse transcriptase. You've got that enzyme, if that gene is still functional, it encodes reverse transcriptase, and it can take an RNA that maybe is just a regular messenger RNA for some regular gene product, it encodes a protein, but instead of just being translated to give a protein product, reverse transcriptase gets hold of it, it gets converted to double-stranded DNA, and that integrates into the genome, and we have what's called a retrogene. You'll recognize these because, for example, they don't have those introns anymore. Remember, the mature messenger RNA had those spliced out. So when you find a gene that has some hallmarks like no introns, and also some other characteristics I'm not going to take time to talk about, you kind of are suspicious that you're looking at a gene that originated through this retrotransposition type of duplication. So there's a lot of families of genes that we are very confident arose through gene duplication and divergence of members of the family. Another classic example, of course, would be the genes for globin, right? So hemoglobin carries oxygen in our blood, and when you're, again, talking now about pregnancy, the fetus needs to get some of that oxygen from the maternal blood, and so it's not surprising that that fetal hemoglobin needs to be a little different, to have a little more affinity for the oxygen than the, say, adult globins. But it's pretty clear all of these globin genes have evolved through this kind of duplication and divergence mechanism. Another example I want to give you is opsin. All right, so the ability to see different colors. The opsin receptors for different wavelengths of light in our eyes and our retina. Well, the ancestral opsin gene is duplicated, and we now have next to each other a middle wavelength and a longer wavelength that can sense different wavelengths, different colors of light. And then here's another one I want to talk about, where you get what's called a chimeric gene. A gene that's got parts from what were originally two different genes. Could be happening at the DNA level. All right, so two different parts, or maybe entire genes in some cases, are brought together, and now they're going to be transcribed as a unit, and you get what were exons of separate genes now being exons of a single gene. Or you can have read-through transcription. The genes don't change their location, but some things happen to make the transcription go farther and read across what was two separate genes. That primary transcript can be spliced to give you a mature messenger RNA that has parts from those two separate genes. And if it gets reverse transcribed, well, you could also get one of those retrogenes, from it. Fascinating example I want to share with you. You might have heard about this one. When Antarctica split off from the bottom of South America millions of years ago, the waters around there became much colder. You had this constant rotating rotating current going around the South Pole, or around Antarctica, and the water got colder, and fish that had initially been adapted to a warmer kind of water now had to deal with water that was so cold that their own blood and body fluids would freeze if they didn't have antifreeze of some kind. And there are lots of organisms, lots of animals and even plants with antifreeze of some kind. Sometimes it's small molecules like glycerol or something, but there are also proteins that can prevent ice crystals from forming in a damaging way. And these antifreeze proteins have evolved in invertebrates, including these fish. There's actually several different examples and types of antifreeze proteins. The one we're looking at here is in a notothenoid fish from Antarctica. And it's called a antifreeze glycoprotein. It's got some sugar groups on the protein. And it's derived from a totally different kind of gene. Trypsinogen is an enzyme, and the gene that encodes that is the ancestor, the original master, that got duplicated, sort of, but it got changed so much, it almost is more like a de novo type gene origination, because let me show you what's going on here. Okay, here we're looking at exon 1, the first intron 1, and exon 2. Now remember, introns are the part of a gene or the primary transcript. At the RNA level, the transcript level, we're going to get cut out so that the flanking exons can be joined together. Normally, that intronic material is not there to be translated into protein. But in this case, the tail end of this intron 1, ACAG, those italicized there, that, as well as the first nucleotides of the exon 2, is now appearing in this antifreeze protein gene. Again and again and again. Repeated again and again and again. About 9 times, I think. One, two, three, or whatever that is. You know, about a little over a dozen times maybe. And that array, that short little thing, is a small, I guess you call it a peptide, oligopeptide maybe, because you get a polyprotein that's much longer than that, and it's got actually 41 of these things in it, and there's a little 3 amino acid point there that's going to be cleaved by another enzyme to break it up into all these little pieces. It's these little short pieces that act as the antifreeze. That's what's going to make it possible for these fish to live at such cold temperatures. Here's another picture showing that. All right, so maybe it's a little easier to see. There's the little part of that intron, normally not encoding any kind of protein. And is taking that and a little bit of this exon, that being then duplicated 41 times in tandem, and that's going to be now encoding this polyprotein that provides the ability to make all these little oligopeptides that act as antifreeze. Now, what's interesting is that a very similar kind of protein, or polyprotein, you find near the North Pole, in the Arctic calm. But it did not come from trypsinogen. It's an example of convergent evolution. And if you look at the DNA, rather than the amino acid or protein sequence, you can see very clearly that it has a totally different origin, and yet clearly the same little kind of stretch of amino acids with a lot of alanines and threonines. Those 2 amino acids in particular are very good at preventing damaging ice crystals. I mentioned that these glycoproteins that we found in those notothenoid fish in the Arctic cod are not the only kind. So that kind is up here, but then there's also some other kinds, including these alpha helical ones. And again, with the alpha helical ones, the so-called type 1 antifreeze proteins, you'll find convergent evolution, similar amino acid sequences because of a selection for a particular kind of function, in this case preventing freezing. Some of the other kinds of antifreeze proteins you find in various fish are derived from other types of proteins called lectins or other sources. So there's a lot of different ways that evolution is found to solve the same problem. Now let's take a look at some genes that encode functional RNAs. Not RNAs that are going to be translated, like a messenger RNA to make a protein, but rather the RNA itself is going to do a job. It's functional. Sometimes they evolve from a formerly protein-coding gene, and sometimes they can evolve de novo from non-genic DNA. Now, the example of a functional, long, non-coding RNA, non-coding in the sense of not coding for protein, I'm going to use is this XIST. Some of you may know that in mammals like us, the males have an X and a Y, females have two X's. And that double dose of X in the female, compared to the male, needs to be dealt with by some kind of what's called dosage compensation. Otherwise, having twice as much expression of X genes compared in the female compared to the male, it's just not going to work. There's a lot of genes on the X chromosome that does a lot of important things, and you need to have some equality of dosage of those genes. So what happens is, as many of you know, is that when a human or similar species female early in embryotic development, one of the two X's is inactivated. It becomes what's called a bar body. It's very condensed, kind of shut down as far as any expression. And it's interesting, I guess there's a kind of hedging of the bets, so to speak. A human female is a mosaic. About 50% of her cells are expressing the maternal X and not the paternal, and the other half the opposite. All right? But in each cell, only one of the X's is being expressed. The mechanisms that kind of inactivates one of the two X's involves this long non-coding RNA we call XIST. That gene has evolved from a previously protein-coding gene that you see still doing that job in chicken called LNX3. And so if you look at all these mammals, there's no LNX3 gene, but what was the LNX3 gene is this XIST gene, whereas in some other mammals and other vertebrates, it's still an LNX3 and not XIST. Some of these other organisms, by the way, they don't have the same kind of chromosomal-based sex determination, so they don't have that same issue of needing to inactivate next. Finally, how about making a gene from scratch, so to speak? Not starting from some other kind of gene or a little, non-coding part of a gene, but truly just kind of from scratch. Well, I'm not sure if what I'm going to talk about now qualifies as completely from scratch. We do have some examples of it really being from just non-coding DNA, as far as we can tell, becoming a gene. But I didn't want to talk about them. I looked, and there's some interesting examples. We think there's probably about 300 of these, for example, that distinguish humans from chimps. But they're so early in their discovery, we don't yet know what their functions are. I can't tell an interesting story about them, but stay tuned. I think there will be lots of interesting stories over the coming years to come out. But the one I want to finish with, I think is very relevant. There are some genes in our bodies, in our genomes, that we call proto-oncogenes. They serve very important, healthy job, function. But they're kind of like the accelerator for cell growth, division, and proliferation. And if they get out of control, it's kind of like driving a car with a stuck accelerator. now you probably have a tumor and perhaps a malignant tumor. Well, there's a family of proto-oncogenes called the MYC family. And one member of that family is expressed in the neuronal cells. So it's called MYC-N. And turns out that you can take, well, we know that this gene is overactive in human neuroblastoma. And if a baby less than 1 year old develops a neuroblastoma, sometimes it's spontaneously recursive. If a baby over a year old, but still very young, develops neuroblastoma, often it's tougher. It may need to be aggressively treated, and it may or may not be a good outcome. And so there's a lot of research being done on neuroblastoma and this particular gene, MYC-N. Well, what's been puzzling is when you put the human gene, the MYC gene, into a mouse, the details of how that works is such that you can get lots of the MYC-N protein, but somehow it doesn't cause in the mouse a good model that resembles what happens in human neuroblastoma, unless you also put into the mouse a gene that's been recently discovered and named factor. It's kind of like Austerberry from Nebraska. All right, so instead of MYC-N, it's NCYM, I don't know how to call it, NCYM. And it is, used to be thought maybe a non-coding. Now it's been shown that it does have a protein-coding exon here. And so this was published just this last year, 2014. And I think the evidence is quite strong that they have. In the primate lineage, well, actually just in human and chimp, there's been this one base pair deletion. And for those of you who know a little bit about genes, you know what a frame shift is, and you know how a loss of, say, a single base pair can do what we call a frame shift. What it does in this case is it enables a much, like 3 times longer open reading frame, the possibility of making a protein of any significant length, over 100 amino acids, in fact, to occur. And there's a lot of evidence that this functions. I won't go into some of the details other than to say that it looks like the protein product, well, I'll let you look at some of the abstract from the paper itself. This antisense, because it's being read from the other strand of the DNA, all right? So one strand is the template for the regular MYC gene. The opposite strand of DNA, and this is very rare for this to happen, but the opposite strand of the DNA in this case looks like it's coding for this other gene, and that they're calling NCYM. A de novo evolved protein regulating the pathogenesis of human cancers, neuroblastoma. It's only apparently in humans and chimps. And if you look at the actual human neuroblastoma cases, you find that this gene is also being overactive right along with the MYC. And if you, like I said, make that happen in the mouse, you get a disease that is much more acting, behavior reminiscent, as they put it, of human neuroblastomas, including distant metastases. So this might be very helpful in terms of opening up some new avenues for figuring out some better therapies for neuroblastoma. And for those who are into cell signaling, you might know that these proto-oncogenes are often part of signaling pathways, and they've identified a particular. It looks like this NCYM protein is helping to augment and preserve the activity of the MYC, which in this particular context is not a good thing because you're getting too much of that already. patience. I really enjoyed sharing with you some of what I consider interesting stories about new genes being formed from auto parts. And I'd be happy to answer any questions. Thank you.
If you have questions and have a mic near you, please use the mic, push the button. You'll get a green light, and then when you're done talking, turn it off again, and we won't get feedback. Thank you.
Hi, I'll jump in and get started off. My name is Tom Ray. I've got 3 quick questions. First one is about the idea of a gene copying, a copy of a gene being made on the DNA, and that second gene then changing and possibly producing other coding for other proteins or serving some other purpose. Am I right in thinking that that second gene is able to do that because the first gene continues to do its job? Because any change to the first gene would kill the organism, you no longer have that protein being produced.
Exactly. Good point. When you have a duplication like that, it can happen exactly as you described. If the original copy keeps doing what it was originally doing, the new copy is free to do whatever. Now, what often happens is it just sustains mutations it inactivate because it's not being evolutionarily selected for any function. But sometimes what happens is the new gene existing, if it does function, at least initially, like the original gene, well, now both of those genes are not as essential anymore, right? And so they both, or one of them, can be kind of decayed a little bit, some mutations that occur and not be screened out by selection, as much by purifying selection that would normally keep them intact. And so you can have what's called subfunctionalization. It might be that now, after some time, the organism is totally dependent on both of them. It can't do with just one or the other. It needs both of them. But another thing that can happen is called neo-functionalization rather than subfunctionalization, where, as kind of we were looking at, one of the copies, whether it's the new one or the original one, either one, is free to, mutate and become do a totally different job. Neo-functionalization. Yes? Good point.
My next 2 questions, and I'll be quick here. My next 2 questions come from videos I've been watching from the Institute for Creation Research.
Oh, yeah.
I'm assigned down at Offhead, and the base chaplain has been showing these videos for the last several weeks. Today they made an assertion that although it's claimed that 98% of the DNA between humans and chimps, that 98% roughly of the DNA is identical, that in fact it's only about 78%. And my suspicion is that the difference is that they're counting all of the differences in the non-coding DNA. The DNA that's not being selected by evolutionary pressures.
Well, it's actually worse than that. I've looked at that argument also, I don't have time to go into all the details right now, but it's a very severe misrepresentation of the facts. So if you actually look at the DNA sequence of the human and chimp genome, they are that 98+% the same. And I remember, one of those 70-some% numbers being thrown out there in a public post letter. And I looked and looked and looked to find anywhere in the literature, and the only place I could find it was ICR results. And then I think I traced it back to a real crude histocytochemistry measurement of DNA, long before they could even do DNA sequencing. But because it had this number that apparently appealed to them, they're still citing it. I don't want to get started on that. But I'm reading the Reason listserv, and your ability to be there and share some insights in that discussion group, I think is laudable. And many in this room know what I'm talking about. And I think it's great.
I don't know that I'm doing any good to the folks.
Well, I understand.
The talks, but I'm certainly getting education. So.
And I have many, many dear friends, relatives I share with you that desire to be very respectful. And my frustration, I've tried to learn over the years to try to be as diplomatic and all as you are, because that's the way we ought to be. And yet to defend the truth and not allow obfuscation or inaccurate information. Or the thing that bothers me most is if school children or even college students think that there's something suspect about science itself, and they have to pick and choose between being a loyal family member, a church member, or being a scientist. And I think it's tragic, because I don't think that's a choice that has to be made. You know, I find a way to put them together, and I think a lot of people do. But thanks for raising that. I'd be happy to share with you some of the details about that 70%. Other questions? Yeah.
Yeah. Going back to the thing about convergent evolution of the antifreeze proteins between the Arctic and Antarctic fish species, were those actually, this might be a dumb question, but forgive me, were those actually the same end result chemical protein, or were they just proteins of similar function?
The one slide I was showing, convergent evolution in the case of the antifreeze glycoprotein. So the Arctic cod up North Arctic and the metathenoid from the Antarctic. That was a curious case where they had the same kind of polyprotein, like 40-some repeated copies of this little dozen amino acid or so peptide. And those peptides were very similar, rich in alanines and threonines. But the DNA sequence that gets translated to make that was very different. So you may know this part about it. I didn't have time to go into it, but the genetic code is such that you can spell, if you will, the codon for an amino acid is probably not the only codon that will mean or signify that particular amino acid. Only 2 of the 20 amino acids have unique codons that code for them. The other 18 have one or more synonymous codons. And so the fact that there's this ability to code for the same protein sequence with different nucleic acid, DNA and RNA sequences, gives you a whole other layer of information about the evolutionary history. And so in this particular case, we know that the Arctic cod protein, as similar as it is, did not come from a trypsinogen ancestor. Yeah.
So, I'm familiar with repeated or highly repetitive proteins from Huntington disease. Is that the same sort of mechanism that happened in the antifreeze protein?
I think so. That's a very good point. So there are some human diseases where the gene involved, the mutation that causes the disease, is because of an expansion of one of those codons. There's 3 bases in a row that constitute a codon. One of those tends to be duplicated. When the DNA is replicated, probably there's a slippage of the template or something, and you get multiple copies. And once you get quite a number, then you reach the threshold, then it's like a runaway train. It can happen very quickly. So in some of these diseases, the age of onset of the disease down to the generation starts happening earlier and earlier, because that gene is more and more mutated. It's got bigger and bigger expansion to it. And if I recall, I think Huntington's, if I'm right, is an example like that, a triplet expansion. And indeed, if you look at that little sequence that got repeated, they're thinking the same thing happened. It was that kind of replication error, template slippage, and then once it got up to a certain threshold, then you can have unequal recombination coming in and giving you kind of bigger increases in size as well. But of course, in this case, it would turn out to be a very useful, healthy antifreeze function. But yeah, there's a number of, especially neurodegenerative diseases for some reason. I guess it's probably because when you get these extra glutamic acid or other amino acids resulting, maybe they cause plaques or fibrils or something to form in the neurons, I suppose. It's not my area, but yeah, they do think it's a similar story. Good question. Other questions? Yes?
Yes, I apologize, I missed the beginning of the presentation. But I'd be curious, have animals such as the naked mole rat had their genes sequenced, given that it's an animal that is reputed to have a very low incidence of cancer, in order to see how it's able to maintain the stability of its encoding?
Well, that's a great question. I love that question, and I have no good answer. Maybe someone in the room does. Yeah. But
I remember doing some reading on that, and that the naked mole rats have got, they're set up for, I think it's apoptosis, such that.
oh yeah, sometimes cell death.
Right, but such that there have to be two mutations in order for its apoptosis not to work correctly, not to happen. So
That's fascinating. Now that reminds me of one thing. I mean, if you compare, I talk about those tumor suppressor genes. So we live a longer life, more chances for mutations to accumulate. It makes sense that we've got kind of redundancy in our tumor suppressor. So two big ones are the retinoblastoma and the p53. And rodents like mice and rats typically only might have one, but not both. But maybe these naked mole rats, even though they're, I think it was rodents, I don't know if they are typically. But anyway, maybe they have both. Maybe they're more like the human in safeguarding against that. That's fascinating.
I just checked. The naked mole rat genome has indeed been sequenced, and it is available on the web.
Thank you, Lauren. Great. Other questions? Yeah.
Yeah, I've got to follow up on the Institute of Recreation Research videos that I've been sitting in on. I often get pushback when I try to point out that intelligent design advocates and these researchers and the folks who are trying to do science toward that goal. When I point out that none of it gets published in reputable journals, that it's not being taken seriously by the scientific community, the pushback I get is, well, of course they don't, because they're predominantly atheists and have a worldview that doesn't allow for any supernatural. So of course we're getting, their opinion seems to be that they're getting laughed out of the room. Their claims and concerns are being taken seriously. I'm not sure how to respond to that.
Here's what I recommend. And I guess you'll forgive me. You may notice on most of these slides, I stated the name of the journal, maybe even the title of the article. This slide, the figure at the top, comes from an article by Graham Finley. And PSCF stands for a journal of an organization I belong to, of American Scientific Affiliation, which doesn't really tell you much, I guess. But that is an organization of Christians who are scientists. And that PSCF stands for Perspectives on Science and Christian Faith. And this article, published in 2008, laid out some examples, some of which I, as you see, used myself, although I typically went to the original articles for color diagrams and such. But the point of this article was to say that, in his view, and I'd say I'd share it, divine providence can use random processing. And from a scientific perspective, we're talking about random processing. And from a faith perspective, that doesn't necessarily mean, in metaphysics, that there's no creator, right? I mean, I share Graham Finley's view. I know a lot of folks in this room wouldn't, and that's fine too. But if you want to provide something to anybody who might have the ears to listen, just write down Graham Finley's name, Perspectives on Science, Christian Faith, 60, page 103, published in 2008. It's available free on the web, and lots of other stuff too. And then if you want to see where I try to collect a lot of links, arguing for this trying to defend the teaching of science, you know, and yet do it in a way that can be embraced no matter what someone's religious and non-religious views are. You know, Roy mentioned some others and I co-founded something called the Nebraska Religious Coalition for Science. So if you go to nrcse, Creighton.edu, that also has a bunch of stuff. Creighton kindly hosts the website. Good question. Any other questions?
Just a very short, simple one. Was that the final slide from car range?
You know, this is, that final slide is on top of a junkyard, or a used car lot, a used car parts place in, I think, California.